We're very pleased that Dr. Matteo Garbelotto will be here today speaking to us about sudden oak death. Some of us here on this call live in the San Francisco Bay Area. Some of us may have oaks on our property. I certainly do. And I think all of us are interested in preserving our beloved oaks. So Dr. Garbelotto has been a Venetian by birth, and he spent most of his life in California, but he grew up in the Dolomites. I hope I'm saying that right. Is that right? Um, he is now a researcher at UC Berkeley and has been there since 1990, and he has been in faculty there since 2000. He created and directed the UC Berkeley Forest Pathology and Mycology Lab, a research center that offers diagnostic services and applied solutions to protect both urban and wild forests in California. He is an advisor to several governments, and he is a world expert on invasive plant pathogens. He is a Fulbright scholar, officer of the International Union of Forest Research Associations, editor on the Australasian Journal of Plant Pathology, and I can't say this next word, Phytopathological Mediterranea. And since 2000, he has been the editor-in-chief of the Journal of Plant Pathology. He divides his time between basic research, applied research, outreach and extension, and also citizen science. He has created and leads the SOD blitzes, a large-scale volunteer-based survey of sudden oak death across California. And currently his main research interests include tree pathogen molecular interactions and the intersection between climate change and tree diseases. So we're really, really happy he joined us today. I have to say I have some personal experience with this because where I live, Los Altos Hills we do participate in the sod blitz, and I had the pleasure of collecting a leaf myself from one of my toy-ons, sending it in, and I was pleased that it was not infected with sod. That was really a relief. And so I would encourage anyone in the future, after you listen to him, think about what you'll do yourself if you have oaks near you or on your property. So Dr. Garbelotto will speak for almost an hour, maybe 50 minutes. And then after that, you'll have a chance to ask all your questions. As he speaks, feel free to put all of your questions into the Q&A, and we will look at them and go in order of received when it comes to the Q&A. So with that, I want to bring you Dr. Matteo Garbelotto. And Karen, I think we may get.
Okay, yeah. Hello, everybody. Thanks for the invitation and thanks for the nice presentation. The only thing I wanted to add to that very, very thorough little blurb about what I do is that I'm also the tree pathology specialist for the entire UC system. So that means that my clientele spans from the Oregon to the Mexican border, besides being a professor at Berkeley. So I have two jobs. And the first job, which is really my main job, to be responsible for large tree health issues in the state, keeps me very busy, as you can imagine, especially with the changing climate. Also wanted to mention that due to federal cutting, my lab is being decimated. And so starting from July 1st, we'll be downsized to minimum level. We'll still be open and we'll still run the blitzes, but not the way that we did until last year. And I'm sure you understand why and what's going on. Of course, my lab is one of the most diverse on campus. We speak multiple languages, and the government doesn't like that. Almost everybody is a woman, and the government doesn't like that. And most importantly, we work with climate change because right now, including sudden death, a lot of the diseases intersect with climate change in different ways. Today, though, I'm not going to talk about climate change per se, although it will come out in the presentation, but I'll talk about sudden death. And I think most of you probably remember sudden death. It's not as popular as it used to be. It's not in the media every day on the headlines, but it's still with us and it's not going to go anywhere. And what many of you may not know, that actually the pathogen was introduced a number of times. And sometimes the actual organism that was introduced was not the same as the one that had been introduced previously. So we actually have multiple organisms causing one disease, a sudden death, but each one of these organisms is different. And the focus of this talk is mostly on that, but I will also talk about the general impact of the disease without going into that detail all the time. So why do we care about sudden death? Well, for those who live in the Bay Area, most of the damage occurred in the past 25 years. The organism, in my lab, we reconstructed the timing of the introduction, and it seems to be in the late 1980s. Since then, more than 50 million oaks have been killed, and more than 100 million trees have been infected. So if you think that the population of California is less than 40 million or about 40 millions, this disease has killed every single individual in the state if we equate trees to people. So it's pretty major. And here you can see two pictures. And as you can see, one of the things to understand is that even if every single tree you see in these pictures can be infected, as you can tell, only some tree species really die in very large numbers. And these species are tan oaks, number one, and then most oaks in the red group of oaks, which includes coast live oak, sheep's oak, and California black oak. So unfortunately, the fact that the disease can infect, the organism can infect any tree species growing on the coast of California, and then it is lethal on some species, creates a very explosive combination in terms of impact, even if it doesn't kill all of the trees in a forest, because as I said, some species are not lethally affected. So many people think that a fire is worse than a sudden death, but the truth is sudden death never goes away. So it will stay in these forest stands forever, and it will make it impossible for a large number of trees, of oaks and tan oaks, to grow in these sites. So why so much mortality? The answer is actually quite complex. And the first way to look at this is by reminding everybody that the organism that causes sudden death, actually I should say organisms now, generically known as Phytophthora ramorum, was not native to California. So in the late 80s, when the first introduction in the wild happened, the vegetation of California had never met this organism. And so as we know, when organisms encounter diseases that they've never seen before, think about measles in Native Americans, for instance, those diseases can be extremely aggressive. And that's for sure the case for sudden death. However, there is a lot more to the mortality than just the plants being very susceptible. Mostly it's the climate that's driving the disease. And so it happens that serendipitously, the climate of the coast of California is basically perfect for the organism. The organism that causes the disease comes from Asia, from the mountains of Asia, and I've been to one such site. And the temperatures at that very high elevation are pretty cool. And there's a lot of relative humidity, a lot of fog in the morning, and then occasionally you have extremely heavy rains, which pretty much matches the climate of the coast of California. But until now, it doesn't really match the climate of the interior of California. And as you can see there are actually 3 different organisms that cause the disease. You should think of these as the organisms that cause hepatitis A, B, and C. So these are actually very different, okay? They are genetically different, they're evolutionarily different. They kind of look the same. And so I, as one of the people who discovered the organism, was not really interested in splitting things up at that time. And maybe that was a mistake. We really wanted the government to control the organism. But so it happens that the government only controlled the NA1 organism in the forest. It didn't control the NA2 and the EU1. Obviously, given that the mortality infection rate is so high, with that loss of tan oaks up to 100% of adults. So in many sites, we completely lost tan oaks, not everywhere. So the species is still alive, but many sites have seen disappearance of adult tan oaks. And we've had up to 80% oak mortality. So obviously, even if not disappeared, in many sites, oaks have decreased their relevance and they become insignificant in terms of numbers. And it's a pity because, as I say here in this point, that has all these little sub-points, oaks are keystone species. So that means that a single tree supports a very large amount of wildlife, a large amount of microbial life, and a large amount of the flora, which means that a lot of the plants and the animals that grow with oaks cannot really survive in optimal conditions without the presence of oaks and tan oaks. But there are other things that are problematic about sudden oak death. One of them is that trees that are infected by sudden oak death, oaks and tan oaks, not about the other hosts, after about a year, even if they look green, their relative humidity and water potential is really low. So the trees is still surviving because these tree species are troopers. They do really well in harsh conditions. But the trees that are green and with a small percentage of water, small water content, are particularly flammable. And so we studied some fires, and the situation was such that any homes next to an oak infected by sudden oak death potentially would burn very easily. And firemen that are familiar with sudden oak death, they don't even go close to a tree that's infected by sudden oak death. The second reason why sudden oak death is problematic in urban areas is because the trees that are infected, they still look green, but they're on their path to death. They fail even when they're green. And that's due to the fact that the moisture content in the xylem changes. There is more air coming in because the cambium is being killed progressively by Phytophthora ramorum. And so these trees become susceptible to wood decay agents, even if the trees are still alive, which is very unusual in general. So that's a problem because you may have a tree that's green, it's infected by sudden oak death, it may fall on you, even if it looks perfectly healthy. The sudden oak death compromises one of the largest carbon offset programs in the world. I don't know if you're familiar with this, but this is a program where you can, if you use a lot of carbon, you can actually buy carbon and you can store it in the land. And a large part of the California Carbon Offset Program is tan oaks, which, as we know, and I mentioned previously, are incredibly susceptible to sudden oak death. Tan oaks are also among the most sacred trees for Native Californians. So it's a cultural and societal huge loss for Native people of California. And of course, we all love how oaks look. And so it's obviously a big loss. Bottom line, the problem is that sudden oak death, it's not just a small problem, it's a huge problem. 150 million trees minimum infected, over 50 million killed. And it goes from the San Luis Obispo area border all the way to the southernmost county in Oregon. So it has a huge distribution that would include at least 4 or 5 states on the East Coast, just to give you an idea of how widespread this problem is. And until recently, we thought that in the forest, we only had one of these 3 pathogens. And so I'll start by that. So that's the one that was first isolated in California when trees were dying in large numbers. Starting in 1995, the mortality became really impressive. Nobody knew what was causing it, and it didn't make any sense because the symptoms were very bizarre, very, very, very odd. And so we found out that the disease that was killing all of these oak trees had arrived in California from somewhere else. Now we know it's Asia, but we didn't know at that time. It was an organism known to man, so unknown to man, so nobody has ever isolated, nobody had ever laid eyes on this organism. And we use very high-tech genetic analysis comparable to what's used in murder cases to identify a culprit using genetic information from saliva or from the blood. And by doing a very similar analysis, we were able to show, as you can see here on the right-hand side, these are the symbols that you see in the square on the right. These are all different populations from different parts of the state. This was a huge amount of work in the forest with a lot of poison oak, collecting all of just thousands of isolates. And when we tried to reconstruct the family tree, so where did these populations come from? And you can see here on top, there is an ornamental plant. It's a rhododendron in this case, but there were several other ones that were very key. So no matter how we mix the data, the population in ornamental plants was always the one that started the entire epidemic. So the ornamental industry really fought this very hard. But when I published this paper, it was the third of a series of papers. They had to accept that they had in fact introduced the organism, and over 125 ornamental plants can actually carry the disease. And the same thing happened in Europe as well, where a different subpathogen was introduced, but we also have that pathogen in California nurseries. So when I was studying Phytophthora ramorum, but we'll get to the point in a second, I was really surprised by how fast apparently it moved, because I discovered it with DeVriso in 2020, in 2000, and a year later already we could find it. Now that we knew what we were looking for, we could find it over hundreds of miles. And so there were two possible interpretations where nobody has ever seen this before. So maybe the organism moves really fast. And we tested that hypothesis by using, again, genetic information. And what you see here on the left is the amount of genetic relatedness in collections of the disease from different points at different distances. So as I move right from the vertical axis, you can see the red line goes down. That red line means that the same genes are not found very often as we move even a few feet away from a tree where the disease is present. So if you want to know how far Norwegian people or Swedish people move, you can see how many people have blue eyes, and you can see people with blue eyes move all over the place. So you know that people are moving relatively far. In this case, we use multiple traits, genetic, not phenotypic, and the fact that we couldn't find the same traits even a few feet away from a source told us that the pathogen was now moving very fast. So when we saw hundreds of miles of coastline being affected by the disease, it was not because the pathogen was moving rapidly. Instead, on the other side of the slide, you can see all those little dots, and there's many more of these little dots. These are all locations where we were able to prove the ornamental plants had been sold or grown, and the sudden death pathogen had escaped from these plants that were ornamental plants that were infected. And because you can see the pink dots are a little bit everywhere, every single introduction starting a little outbreak, and when you add up all of the outbreaks, you get to hundreds of miles. So this is nice. It's a good story. We were able to prove it with very strong genetic data. But how do we explain the movement of the pathogen in wildlands where there are no ornamentals? I mean, ornamentals may be here and there in private properties, but it doesn't really explain how the pathogen really could colonize. We're talking, millions of acres of land. Serendipitously, we discovered that in California, this oak killer doesn't actually spread on oaks at all. So oaks are not infectious. What is really, really infectious are the leaves of California bay laurels. And so this is the first time in the history of plant pathology that we have a disease that really sporulates on one host, but then kills another host at this scale. And these are diseases that are very well known in humans, and we call them zoonotic diseases. These are diseases that are spread by vectors like mosquitoes, and then they infect dead-end hosts like humans or horses, like in this case, West Nile virus. And the disease is only expressed on the dead-end host. So oaks are dead-end hosts, and the disease is expressed in them. But bay laurel doesn't really die because it's infected. In fact, all of it has are these blackened tips on the leaves. And if you had supervision, you could see these global football structures that are the infectious structures that move from tree to tree. They move in the air when it's raining because they need droplets of water to move. And the football opens up at the end when it reaches a plant surface with a film of rainwater on top of it. And the smaller spores actually come out of the flying saucer here. You can see them coming out on top of them. They're actually swimming, and they swim into the host. So this is unfortunate because bay laurels are very good vectors of the disease, and they don't die. So actually what happens in areas that have a lot of sudden death, even what the ecology had predicted years ago doesn't happen. So instead of having a shift in many sites from Oakland to Douglas fir, we have a shift from mixed oak bay laurel woodlands to almost pure bay laurel woodlands, which means more sudden oak death will be spread. And when you have a lot of bay laurels, not just oaks become susceptible, but the amount of pathogen in the air is so high that other species can be infected. And in fact, a few years ago, we found out that several rare and threatened species of manzanitas were at risk when they were growing in proximity to bay laurels, because bay laurels are so infectious. So if you want to look at the distribution of sudden death, you can see it here on the right-hand side. So you can see it's really widespread in Northern California, Central California. The darker the color, the more disease we have. And then you can see the distribution of bay laurels on the left. So you can see there's a pretty excellent match between the two. Some areas that have bay laurels don't have the disease.
Turn this down.
It's not really arrived there yet. Okay, so it makes sense that the disease's distribution would be smaller. So I was saying that it's not just a matter of plants being very susceptible, although certainly that's a key, but it's also a matter of whether the conditions are right for infection. And in a series of papers, we have shown that the conditions on the coast of California are basically perfect. On the left-hand side is the paper where we first reported the bay laurels were really drivers of the disease. And we also found out that the pathogen itself is moved around by at least 50 species of plants, including, for instance, the Western starflower. Just those species don't produce enough of the pathogen in terms of spores to cause oak infection, but they can definitely cause oak movement. And the most famous of these hosts is redwood. So redwood can actually move the pathogen without causing any mortality until the pathogen arrives to a patch of bay laurels or tan oaks. And then where the patch is, you have a buildup of the pathogen and you have mortality. The other second paper published here was about the temperatures that are ideal. And you can see that about 69 Fahrenheit or about 20 or 19 Celsius are ideal. And that temperature is on the coast of California is very, very common. As you see, as the temperature goes towards 90, it becomes really less favorable. I'm sorry, I'm here in the middle panel, and the temperature is actually the lower panel of the middle figure. So you can see the higher the line is, the more favorable the conditions are. So you can see that at 15 Celsius, they're okay. They get really great at 19, 20. And then as temperature warms up, the conditions become less favorable to infection. And at the same time, on top of that, we showed that you need to have leaf wetness for at least 12 hours to really get a good level of infection. And on the coast of California, where the temperatures are cool, it's very common that leaf wetness will persist for at least 12 hours. And then finally, on the last panel on the right-hand side is how and when are oaks susceptible. And what you see here is there are two graphs. You can see on the top graph, when you see a peak, it's when bay laurels produce the most spores. So that's when they're most infectious. On the bottom panel, you have when oaks are most susceptible. You can see that when the bay laurels are most infectious, what happens to oaks? Oaks are most susceptible. That's serendipitous. It's just a coincidence, but it's very unfortunate. Because if the two cycles were somewhat off, then we wouldn't have so much infection of oaks as we have witnessed in the past. So again, we call it synchronicity. Synchronicity between infectious activity of the pathogen and susceptibility of the host. And this is not a given, okay? In many cases, you don't have synchronicity. Finally, the last piece of information is in order to get oak infection, because really, if we didn't have oak infection and tan oak infection, I don't think that sudden death would be such a big deal. Just a disease of leaves of bay laurels or needles of redwoods without really causing much disease, it wouldn't be such a big deal. So really, it is oak mortality and tan oak mortality that are big deals. And when do we have oak mortality and tan oak mortality? Well, oak mortality in particular doesn't happen every year. And this is really bizarre. It's actually difficult to infect an oak. We have problems infecting oaks in the lab, but the disease does a great job of infecting oaks, and it does so when two prerequisites are met. The first one, the oak has to grow next to a bay laurel, And the second one, infection happens only when we have extremely wet years, like El Nino, for example. So here in this graph, I show you three things. I'll show you rainfall levels in blue. I show you how much sudden death there is, active sudden death that we can actually culture and test. So you see there's a perfect, almost perfect relationship between the amount of water and the amount of sudden death. And when you reach peaks of sudden death, then you actually get oak infection. So oak infection doesn't happen every year. It happens every 4 or 5 years, which if you are a resident of California, you know that it's kind of the cycle of either the Nino or it's the cycle of wet years in the state. So this has been problematic because when oaks don't become infected every year, people assume that their oaks are safe, but in reality, they're looking at the oaks during the dry years, far away from the infection peaks. So the infection peaks happen when it rains a lot, much more than the average of the 30-year average. Mortality of those oaks will happen between 1 and 5 years after infection. So you see tree dying during the dry season, but they're dying just because the disease is developing, because no disease is immediate. But all infection happened only on those where those arrows are shown since we've been studying the disease. So again, only 4 times in the last about 15 years or so. So what can you do? So remember, there's 2 prerequisites for oak infection. And the first prerequisite is how close it's growing to a bay laurel. And here on the left-hand side, this is a huge study that we did in the peninsula, looking at what the likelihood of oak infection was. And those are those lines that you see with those colors as you move away from a bay laurel. So the vertical axis is a bay laurel. And then these are meters that you're moving away from a bay laurel. So 5 meters, 10 meters, 15 meters, and 20 meters. So if an oak grows 20 meters from a bay laurel, would you think that the chances of infections are high or low? So the chances of infection are expressed on the y-axis. The higher the lines are, the more risk there is. So as you can see, as you move away from a bay laurel, the risk of infection for oak basically becomes asymptotic to 0. So there is really no risk for oaks that grow away. I mean, these are large numbers. We were monitoring over 3,000 oaks for over 7 years. And these are the results. So as a result of that study, first of all, we really tell people that if you can clear your bay laurels from about 10 meters around your trees, your infection chance goes down really dramatically. And sometimes those bay laurels are very tiny bay laurels that are growing next to a gigantic oak. So it's a strategy that makes some kind of sense, but we had to test it. And here we are testing it. And so we had 4 large plots in the Santa Cruz Mountains. And we had in 2 of these plots, we removed all bay laurels within 10 meters. And in the other plot, we didn't do anything. When you look at this graph on the right-hand side of the slide, you can see the 2 different treatments as different colors. The green color is the one where the bay laurels were removed, and the bay laurels were removed when the dotted line, you see there's a vertical dotted line? That's the year and date when the bay laurels were removed. And you can see, and the higher the line is, the more likely the chances of oak infection is. It's actually the number of places where oaks can become infected. So you can see that when we remove bay laurels, the line stays very low, meaning that a very low number of oaks will become infected. If you don't remove bay laurels, you can see that the number can go very high, especially when it rains a lot. And also you can see that when you have a drought, you basically have no oak infection. So that's 207, 208, 0, whether it doesn't matter whether the bay laurels are there or not. It's too dry for the bay laurels to produce enough spores to actually infect an oak. Bay removal near oaks is key. Actually, I should have put other pictures that you may be interested in. The other key, if you're a land manager, like say a large land manager like Mid-Peninsula Open Space or the San Francisco Public Utilities Commission, you may want broader management approaches, and simply lowering the density of bay laurel in general lowers the disease. Just if you use that approach, you won't know which oaks will become infected. So for large landowners, it makes sense to just lower the abundance of bay laurels. that will reduce the overall number of oaks that are killed. If you're a private owner and you want to protect specific oaks, it makes sense to remove bay laurels that are next to your oak. I mean, in the end, you're doing the same thing, but if you're a private landowner, you're more targeted because you want to save those oaks that you really care the most. If you're large landowners, you just want to save oaks in general, and you can achieve that by lowering the density of bay laurel. By the way, we're not afraid of saying this because right now, based on research that was done in the 70s at Berkeley and then continued in the 80s into the 2000s, we have the amount of bay laurels in California is 3 times as much as what it should be because of fire exclusion. So when you're removing bay laurels, you're actually bringing the system back to some sort of a balance that it should have. So fire exclusion and grazing practices, and I cannot get into detail, has allowed bay laurels to grow exponentially 3 times as much in just about 40 years. So basically, by excluding fire and by allowing grazing, we have created an ecosystem where the vector for sudden oak death is super abundant. And as a result of that, we have these very high levels of oak mortality because the vector is basically everywhere. If you have a monumental oak, we also developed really quickly benign chemical treatment that increases the immune response of oaks. So this doesn't really do anything to any other organisms. It does repress phytophthora from growing. Almost all phytophthora are detrimental. And most importantly, most of the action is obtained by increasing the defense response in the oak. And you can see these are some of the data that came out. We produced quite a lot of data. The high bars means a lot of disease. It's like the lesion, it's like the amount of cancer in a person. So that's a canker size in a plant. And then you can see how significantly lower the amount is once you use this compound, which it's called phosphonic acid. So we really combine the two. So now, if you were following me, you know that sudden oak death doesn't move very far. So a single bay will, basically cause disease around it for about 30 feet, 40 feet or so. And then, of course, the disease can go from bay to bay. But understanding where the infection is makes a huge difference if you are any kind of landowner. Because if you are a mile away, it may take years for the pathogen to arrive there. And so given that from the beginning, the distribution of the disease went from San Luis Obispo to Oregon, we absolutely had no resources to map all this huge amount of land. As you know, the mountain ranges are very rugged and so very difficult topography. And so we had this idea of recruiting people in local communities. And we started what has become now one of the largest citizen science programs in the world, targeting infectious diseases of trees. It's a very serious program. So volunteers do have to go through a training program. When COVID hit, we were the only large-scale citizen science program allowed to continue because we were able to go contactless within 2 weeks. So all the training can be done online. You have to register online. You can only participate if you've done the training. You tell us where trees have symptoms. You collect leaves, bay laurel leaves, where you see the symptoms. You tell us where the trees are. We do all of the testing at Berkeley. So it's a hybrid format where the volunteers tell us what's on the ground, and Berkeley confirms the presence of the disease using DNA tests, the DNA tools, because visually you can never be sure because there's many things that look alike. So people sometimes tell me, oh, I know I have it. I know I have it. No, you don't have it. So what we do with this program, we survey almost 20,000 trees per year. And we test 2,000 trees per year from all different communities. And we post the results in real time. And these results are available online. You go to sodmap.org and you can actually download an interactive map that tells you where sudden oak death has been found. If you want to see the last year's results, you go to the SodBlitz map, again, sodblitz.org. If you're a scientist, you will go to Cal Invasives, which is run by CalFlora. So CalInvasives allows you to map invasive pests and pathogens on in California. And if you have an iPhone, you can also use SodMap Mobile to see where infections are. We have over 50,000 points now after 18 years of the program and 70 years of collection. Knowing what the pathogen is makes a big difference. Again, if the pathogen is in your township, but it's miles away from your property, you really don't have to worry. As the pathogen gets closer, you can see it on the map getting closer. Then you can start thinking of whether you want to remove bay laurels and whether you want to treat some of your most valuable oak trees. But let's go back to the different pathogen causing the disease, and I'll tell you now why why it matters which one. So we actually have 3 different pathogens that cause the same disease. They haven't seen each other for about 500,000 years. So you can imagine what happens in 500,000 years, considering that this pathogen can complete its life cycle in a day. So it's a lot of life cycles. And as a result of that, these 3 groups of the pathogen have really diverged significantly. They cause disease in different, with different severity. They also have different temperature requirements, and they sporulate in different ways, but they cause the same disease. And originally they were the same species, but they're not anymore, but the taxonomy hasn't followed up yet. All 3 of these were discovered in my lab. In 2001 or 2002, we discovered we had 3, but we were not so worried about them because the 3 pathogens were only present in the ornamental industry. And in the forest, we only had one, the one that's called NA1. So NA1 was the one that was present everywhere. A1 and Na2 were only present in the ornamental industry, and I don't work really with ornamentals per se. So we really focused on the Na1, and every 20 years of studies were done on the Na1. One of the things, we learned about Na1 was that Na1 is rapidly evolving since it arrived in California. And one of the things that seems to be triggering its evolution, or actually slowing down its evolution, it's temperature. So when we have higher temperatures, like the temperatures in the interior of California, we have noticed that the evolution of the pathogen is slower because the pathogen the NA1 pathogen doesn't really like the warmer temperatures. And here we have actually a graph of California and the different hatching that you see. It's actually different genotypes that have evolved. These that you see here, they're actually like variants. If you're familiar with the COVID variant, this is the same thing for sudden oak death. But these are all variants of one of the 3 pathogens, okay? But as you can see, they have a different distribution. And sure enough, the ones that are up north do better in the cooler climate, and the ones that are south do better in the warmer climate. So there is some sort of adaptation. So the pathogen is smart. It gets selected when it does very well in a certain environment. Okay, so do we have resistance in our hosts? We don't really truly, I'm not sure there is any resistance. We do have some tolerance. So some trees will take a much longer time to die, but eventually they will die. And some trees and some oaks, actually, they appear maybe to have resistance, but nobody has really studied how that works. We studied tan oaks, so we know how the trait is inherited. And so we know that some mothers produce acorns that are much more tolerant to the disease. And when I say much more tolerant, we've measured up to 20 times more tolerance, which is a lot, which means that if you use tolerant plants and you space the plants out, remember, this is an infectious disease. So all of it spread has to do with social contact. So if you reduce the social contact among tan oaks and you use more resistant germplasm, these tan oaks will actually have a much, much higher chance of survival. And we worked with some native First Nations to prove it, and we're in the process of proving it, but it's a very long-term study. Sorry, do you want to go here? Okay, so until 2020, we really felt that only the NA1 was present. We were not concerned about the other ones. In 2020, we found the EU1. but we found it in the cornermost northern west corner of California. And we were able to show that, not that it matters, but we were able to show that that pathogen, so the EU1, which is very common in Europe, that's why it's called EU1, had arrived from Europe to North America through ornamental plants, and then from Asia to Europe through ornamental plants. And then infections in Oregon were responsible for the outbreak in the Nordic County. Very, very limited outbreak in the very corner of the state. However, that finding in 2020 broke our assumption that we only had one introduction in the wild. So because all of a sudden we had two, even if the other one is very limited. And so we asked the federal government to give us funds also to test which one of the three pathogen was present. And we were maybe hoping that we wouldn't find it. Remember that even if the USDA has monitored ornamental plants, they were not able to eradicate the disease from the ornamental industry. So even with all the controls, the pathogen is still present. So there was a possible pathway because we knew that all three of these pathogens were present in the ornamental industry. Sure enough, this year in 2024, we found the third one. So we had the NA1 from the beginning. Then in 2020, we got the EU1. And now in the Bay Area, we got the NA2. So why is that important? So I'm going to show you where the NA2 is. This is based on the fall, but we've done some more sampling in the winter, and unfortunately, it's much more widespread. So where is the NA2 here? So the NA2 is the orange trees. And it's not just one tree because this is a large scale, so they're stacked one on top of the other. So in almost every location, we have multiple trees, and now we have miles of trees that are infected by the NA2. The red icons are trees that are infected with the NA1. Okay, so that's kind of the garden variety, we can call it by now. And this is Burlingame Hills, so in the peninsula. So now you can see all the trees, and this is based on last fall. So you can see it's quite widespread. And then this is the largest outbreak, and actually turns out that almost every tree west of 280 is infected by the NA2. Well, if it causes the same disease, here we are, NA1, E1, NA2. This is like a family genealogical tree. So you can see that we have three branches that are clearly distinct. Each one of those is a different lineage. We could call it a different pathogen because they are. Why are we concerned if the disease is the same? Well, we are concerned because the three lineages are different. So it's not just because one is more aggressive than the other, although it is the case. More importantly, they're different. So it means that those trees that escaped NA1, they may not escape NA2 because NA2 is better at doing something. And I can tell you from the beginning that NA2, which is the one we just discovered, does better in drier and warmer weather. So all of a sudden, areas that already are infested by the sudden death pathogen, a lot of microclimates that were warmer, those were spared by the disease. But now with the NA2 can really cover those holes that are drier and especially warmer. So when you have warmer temperatures, you can have NA2. And then as you transition to cooler spots in the same stand, NA1 does very well. So you understand that when you have NA1 and NA2, it leaves very little hope for trees to actually escape infection, because now NA2 is present where NA1 is not present. And so luckily we had done, both at Berkeley and in Corvallis, we've done a series of studies about comparing the different lineages that we had found in the ornamental industry. And you can see the bars are different. So the light bars are the new introduction, the NA2. And you can see that this is how aggressive it is. So you can see that in this particular test, the white bar tends to be more aggressive. Not always, I won't go into all the details. So already we have some information that even in some cases, it's possible that the NA2 may be more aggressive, and the data suggests that. And this is the data looking at temperatures, and I would like for you to just focus on the black bar that you see in these charts. And compare the black bar on the right-hand side with ones on the left-hand side. So if you can see the bars on the right-hand side are higher than the ones on the left side in general. So this suggests that, again, this is how many trees are infected, no, not how large the lesions are. This was lesion size, so how severe the disease was on each individual tree infected. This is how many trees are infected, with one being 100%. And so, as you can see, in a warmer weather, Na2 can infect a larger number of trees, maybe 10, 20% more, which is a lot if you think about it in epidemiological terms. And if we want to look, remember those oval-shaped structure, football-shaped structures that are responsible for infection. Actually, this is lesion size again. I'm going to skip it. I want to show you the number of sporangia that are produced. So look at this graph here. So the higher the number, the more infectious the pathogen is. So you can see that Na1, which is the one that we have everywhere in the forest, is the least infectious compared to the other two. So the other two produce more than double the number of spores, which really means more than double of the trees can be infected. So that means that the speed at which the disease spreads can double when you have Na1, when you have Na2 and E1 in addition to Na1. So all the metrics are there. And then finally, what I wanted to show you is, again, you see the three pathogens here. Na2 is the one that we just discovered in the Bay Area. I wanted you to look at the number of those spores. This is a different way of measuring that, but it's a number of spores that we're seeing in a volume of liquid. You can see that at 80 degrees Fahrenheit, the Na2 is almost 4 times more aggressive than the Na1 in terms of production of spores. You see that 36.4 that you can see here. So we have good reasons to be worried. And here is another study. So these are all different studies by different people, some mine, some are not mine. Look at the Na2 here and look at the Na1. So when we approach 90 degrees, actually 80 something, the Na1 stops growing. You see there's a 0, but look at the Na2. The Na2 still grows quite significantly compared to the Na1. So this is just growth rate. This is just living. The organism just live comfortably. And yes, it can live, maybe not comfortably, but it's living, and quite significantly more than the Na1, which is basically stops growing completely. Doesn't die necessarily, but it stops growing. Not so for the Na2 at this very high temperature. And this changes our predictions of where sudden oak death is going to hit quite significantly. So this you see here are predictions based on Na1. These are based on real data collected by the citizen scientists. So we have here, at the time we did this study, we have 35,000 data points. And we were able to show the bay laurels lose their infection status. So they revert to be healthy by dropping their leaves. And that happens every time that the temperature warms up to a certain level in a site. So the warmer sites see a high incidence of that happening. And this is the orange line, and the red line are the temperatures. And as temperatures go up, you can see the amount of trees that lose infection almost reaches 100%, right, at the edge. So almost every tree that grows in very warm climates, so climates where 90 degrees are reached frequently, they can lose the infection completely. But and then the amount of rainfall also is significant, and the less rainfall, so the blue lines is rainfall. So you see that as you increase the rainfall, the less recovery there is, which means the more disease you have. So now all of a sudden we have to change probably these curves, because the disease can survive at warmer temperatures. And so all of those areas of California, even locally they were spared, are likely not to be spared in the future. And that's why we're so concerned. And then there are other things that we're concerned about, but I can tell you the chemical that I was showing you earlier on, and I was showing how effective it was, well, it's less effective on E1 and Na2. And the reason for that is, and I don't want to go into the details of showing you that, but the reason for that is because Na2 has been exposed to the chemical in the ornamental industry, where plants are treated regularly with insecticides. And so as a result of that exposure, so we published this paper a few years ago, we were able to show that the pathogens that spend a significant amount of time in the ornamental industry become more resistant to the chemical, once they go out in the wild, that resistance remains. And so the chemical could become less useful in the future as Na2 and E1 spread, while it is relatively useful for Na1. Okay, so bottom line, we know now that we have it in 6 areas, 5 in the Bay Area and 1 up north. We've been working mostly in the Bay Area. So what we're doing now, we're creating zones of infestation for the landowner. So these don't have any legal value, but we're telling, if you, for instance, this is the one we created after the first survey. So this includes a 1 kilometer, a little bit over half a mile of buffer zone around the trees. So these are areas where it's possible Na2 may already be present. And the most important BMP is not to remove bay laurel leaves when you do trimming and any sort of forestry or landscape activity, especially, at all from these yellow dots. So they have to stay there. And the second thing that we're also suggesting to remove some of the trees that are infected, but that option is already becoming possible for the first circle on the left. Maybe the first 2 on the left is too much of it, but it may still be viable for the 3 circles on the right. So we did this for every outbreak. Also, if you have a nursery and you're selling plants and you're within one of the circles, it becomes complicated. And the landowners are already telling the nurseries that they need to have their plants checked pretty strictly to make sure that we're not spreading the Na2. Because remember, when the Na2 was spread, it was spread by the ornamental plants over and over again all over the state, and that's why it became so widespread. Okay, so here it's kind of a summary of those. I'm not going to go over all of them, but there's some more detail here. So, one of the things we've been saying is just leave the bay laurel leaves on site. Leave them, don't pile them up, because the foliage at the bottom of the pile remains infectious. Just broadcast them where you near the tree that they came from, and if you can, mix them with some of the topsoil, because the organisms of the topsoil are particularly aggressive, and Phytophthora ramorum is not a very good soil inhabitant anymore. Even if Phytophthora were born as soil-borne organisms, Phytophthora ramorum, it's at the top of the pyramid. It has evolved to be aerial, and as a result of that, the trade-off is that it doesn't do so well in soil. Okay, and so that's why we have shown over and over again that people moving soil, bikes, hikers have not spread sudden oak death in the Bay Area. So sudden oak death spread from ornamental plants multiple times on itself using the air. Okay, so it spreads by itself. But of course, if you bike a lot in Los Altos Hills, Kristen, and then you go to Tahoe, that is different. So you could potentially bring the disease to Tahoe by moving a dirty bike from one location to the other one. But if you bike, all over the peninsula, it's not going to make a difference, because the pathogen really spreads on its own. And I like to finish with 2 slides. Unfortunately, I have the right one here. So the salt blitzes continue in spite of the funding cut, and everybody's we're going like hotcakes, this program right now, because everybody's scared about the Na2. So people want to know where is it so that they can curtail its spread. If you go to saltblitz.org, every year you will find a calendar of blitzes near you, probably for some of you. They are organized everywhere from the Oregon border to Santa Barbara County border on the coast, and you can choose the closest one to you. And you go online and you train. So saltblitz.org, all of the collections and surveys are done in the spring, because in the spring the symptoms become very obvious. Because remember, it needs rainwater to spread, so it's really active in the spring. If you do it in the fall, you miss more than 50% of the trees, because we can't just see the symptoms, okay, on the trees. And if the other thing I wanted to mention is the oakstep.org program. So if you want to go on Oakstep, so if you are, we don't have resources to do oak testing, and very few people in the world really know how to do this oak testing. But if you register for literally for pennies as a license, you need to be a licensed arborist. We train you on how to do your work with the oak. We give you the materials that are necessary, and then you ship us the plates and we do the diagnosis for you. All of this literally almost a very limited cost, about 1/10 of what it would cost you if you used a private lab. So these are the two websites I wanted to end with. Oakstep.org, that's for oaks, and sodblitz.org, that is for bay laurel leaves and for tannock leaves. But remember that it's bay laurels and tannock leaves as well that spread the disease. So you always have infection by sod on bay laurels first and then on oaks. So if you test your bay laurels and you find it, then you know that the next wet year, the chances of oak infection is very high, especially if your oak is within 10 meters from uninfected bay laurel. I am done, and I will, we can, I guess, open the floor to questions, and I can go back to some slides too, but I will for now take this off.
Okay. Thank you so much. I'm thinking of so many questions myself, but I'm going to go with everybody else's. So Lisa Barboza asks, is the canyon live oak affected?
Yeah, so canyon live oak is affected, and it's the most complicated. So basically, black coast live oak, Shreve's oak, and black oak, coast live oak, Shreve's oak. Yeah, those three are pretty much identical in terms of the symptoms they develop. But canyon live oak often develops a branch dieback rather than the entire stem. So, but sometimes also the entire oak dies, but we haven't really been able to identify infections that kill the entire oak. So it's a little bit of a question mark, but yes, it does get infected. While, for instance, blue oak and valley oaks and Oregon oaks do not get infected. So it does get infected, but the outcome of that infection can be variable. So some oaks just lose a branch, and some oaks the entire stem will die.
Thank you. So here's a question from Charles. Dr. Garbelotto, would reintroducing fire under oaks help the situation?
Yeah, so yes, for sure, greatly. And but I'd like to point out that not the way some people think, and some papers have been published which claim this, and we know they're faulty. It's not, the fire doesn't necessarily get rid of the pathogen, but what fire will do will lower the abundance of bay laurel. So it will work a little bit like mosquito abatement, okay? So to be honest, to be totally honest, the year after a fire, we find a lot less sudden oak death in a site. We do. Okay, so it's true that it does affect the pathogen, but only for 1 or 2 years. So after 2 years, the pathogen is back at the same level it was before the fire, very fast, very, very fast. But if you can plan your fire, your prescribed fire, I guess, or whatever you want to do in a way that it lowers the percentage of bay laurels, then you're really protecting a much larger chunk of your oaks, because it's, again, as I said, it's like mosquito abatement. The less mosquitoes you have, the less malaria. The less bay laurels you have, the less disease, the less sudden oak death. The other thing that you should be aware is that I've been studying bay laurels because they're showing up with a lot of diseases right now, not sudden oak death, and we've come to the conclusion that bay laurels that now are growing outside of riparian corridors and outside of draws. So those bay laurels that grow everywhere, because of fire exclusions, they become really susceptible to all sorts of diseases that are triggered by climate. So they are very susceptible because they are in a suboptimal ecological site. So really the point is, let's let the bay laurels be in the riparian corridors, let them be in the draws. And when you add them there, then unfortunately you may not really have a solution for sudden oak death. Maybe that's the place where bay laurel wins and sudden oak death wins. But in those areas that are drier, those are areas that should be mostly populated by oaks. And we need to remove, that's really where we need to remove the bay laurels, because they don't belong. And they're falling ill for a variety of reasons. So ecologically speaking, there is really also double, two birds with a stone kind of thing, where we are restoring the habitat to more, in quotation, in natural conditions.
By the way, can you give us an example of some of those hotter, drier areas in California? What would
But
qualify?
every time they use, okay, bay laurels now grow everywhere, literally.
Mm.
Literally, so if you're not near a riparian corridor, and if you're not in a draw, you're like on the slope and it's exposed to, it has an eastern exposure, or a southern exposure or a western exposure, Bay laurel is just proliferated there because the lack of fire, but it's not really its ecological optimum, you know. I mean, originally bay laurels were not everywhere. So when the Europeans arrived, bay laurels were really associated with shady areas, draws, and riparian corridors.
Okay, great. So here's another question. Are Pacific madrone able to spread the pathogen?
So yes, so those of you that are familiar with Pacific madrone, you know that if you blink too hard, they die. So they're very susceptible to a lot of things, which is unfortunate because they're so beautiful. And you probably are familiar with a canker disease that they get. So they get one canker disease on the branches, and then they get one canker disease on the stem. They're very visible. There are areas that become all black, and then everything above the lesion dies. Okay, so obviously when you get it on the stem, the entire tree dies. When you get it on a branch, then just that branch dies. So mostly Ramorum does branch cankers on madrones. So we haven't seen any, and then it can always obviously kill smaller madrones for sure, but we haven't seen the disease kill entire large madrones. It does sporulate on the leaves, but madrone is so susceptible that basically bursts. You know, the disease becomes really aggressive, but then it kills itself because the leaf dries up really fast. And as the leaf, dries up, the pathogen becomes less abundant as well.
So
So
can they spread them or?
they can spread them. So they're certainly on a radar in terms of number. Maybe I can think of 5 cases where I've seen probably I probably looked at about 10,000 oaks. I can think of 5 where madrone was a possibility. And I can tell you that the same thing applies to Toyon. But Toyon is a little, because Toyon doesn't die as rapidly when infected. I mean, the portion of a Toyon, I think Toyon can be more consequential than Pacific madrone. But both of them can infect other plants, but and sometimes maybe even oaks, but at a very, 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 very low rate. So
Thank you.
yeah.
That's good to know. Okay, so here's another question. Is there a natural predator or a way to something naturally that controls it in its native region?
Well, oh, it's native region, yes. Yeah, that's a study that people have to do, but it's very difficult to get a permit to study in China and Vietnam, and so for sure the condition when I went to the site where the sudden death pathogen comes from, but there's multiple areas where it comes from, and this may explain why we have these 3 different pathogens because each one may come from a different area. So that's it's almost sure, but the one that I visited, the disease was almost undetectable. So we can find a pathogen, but it's really not causing disease. And when I did a study that I never published. Berkeley has the best collection in North America of rhododendron species. Oregon has the best collection of varieties, but we have the best collection of species. And guess what? I found out that rhododendrons that originally come from Bhutan, they were a lot more resistant to sudden death than other rhododendrons. So that's probably another area where the disease is present. And so the local flora has co-evolved to some level of resistance. So I found that very interesting. I should have published it. Maybe it will. Good knows. I don't know. Too long
I think
ago
this
now.
crowd would be interested in it. Okay, so is the European laurel a vector plant?
Yes, it is. But um, probably an order of magnitude less than California bay laurel. So it's not black or white, it's gray. Um, I wouldn't want to have, so that means that the risk zone near a European bay laurel or Italian bay laurel, or Grecian bay laurel, whatever you want to call it, may be smaller. So I think if we tell you 10 meters, 30 feet is a good kind of rule of thumb for medium-sized oak. Maybe for Italian bay laurel, it would be 5 meters, right? So the oak would have to be closer to the Italian bay laurel in order to become infected because the Italian bay laurel doesn't sporulate as prolifically as it does on the California bay laurel. So bottom line is I wouldn't want to have an Italian bay laurel really close to an oak, But if I had Italian bay laurel near the house and I had an oak that's, let's say 40 feet away, I wouldn't worry too much about it.
Okay, good to know. So next question from Karen Mackey. What land use practices promote the proliferation of sod?
Well, so the two main, okay, so the main one is green waste. Okay, green waste is, because remember, oaks are not really infectious, so it doesn't really matter what you do with oaks. Really, the big deal is bay laurel and tan oaks. Okay, so those are the two major spreader, and of course some ornamental plants. So that we should throw those in the mix. So green waste management is kind of key because when you have this, let's say, rhododendron, camellias, pieris, tan oaks, and bay laurels, they can be very, 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 very infectious. And so by moving the foliage around, you also increase the chances of spreading the disease. In particular, if you're doing landscaping or forestry during the rainy season, because that's really when they're most infectious, and at that point, if you're doing major land movement, bulldozing, major, not just one, major land movement, and you're moving large amounts of soil during the rainy season, you may also be moving the pathogen to the site where you're moving the soil to. So that would be the number two thing that you could do at the large scale. So the other thing that it's bizarre, so you got to be careful not to, when you have active sudden of death in bay laurels or in oaks, in both, obviously. Tarping, because of the beetles, we normally like to tarp. The tarping can actually facilitate the spread of Phytophthora under the tarp. So that is an exception to the rule where you shouldn't really be tarping the trees that you cut, especially if you have the foliage in those trees. But here's kind of the weird thing. The other thing that you do normally is to debark trees, again for bark beetles. You take the bark off, and then you just let the bark logs. If you do that, Phytophthora ramorum, the agent that causes sudden death, can actually sporulate on the debarked log if it gets wet enough. So again, you don't debark oaks and tan oaks that you've cut down because they have sudden death. You leave them with bark on, and you try to put them in a place locally with as much light as possible. And the same thing applies to the foliage. And of course, with the foliage, really, you got to be careful not to move it around. If you're chipping, things, what we recommend is that after you chipped foliage of tan oaks and bay laurels, you chip some wood, because the wood doesn't have the spores. So by chipping wood, you're kind of rinsing your chipper. So it's definitely not surgery level practice, but it's the best you can do normally, because you can't really get into a chipper and clean it.
Oh, wow. So all the things people are trying to do for good reasons end up spreading many of these things, it sounds like. Okay, so another question, you had brought up grazing, I believe. Charles Little asks, what role does grazing play?
Yes, yeah, that's a very good question. So when you have grazing in a mixed woodland, cattle just will not eat bay laurel, but it will eat oak leaves. So as a result of that, oak leaves get damaged and bay laurels flourish, because you have a third element here affecting negatively oaks and not touching bay laurels. So because of that, bay laurels become a lot more competitive than oaks.
Okay, good to know. And then many of us are wondering, is there a way to donate to your lab?
Yes, there is. So, and it's totally tax deductible. And I can tell you that the donations that people have made allowed us to keep the lab open between March and June. So it was a huge thing that we could do. So we could actually run the Sublets this year. So for donation, you have to go to Matteo Lab, Matteo with 2 T's, matteolab.org, and then you'll find the donate button.
Okay, and if you can send it to us afterwards? We can also include it with your PDF when we send it out. And
I'll add a slide with the donate button.
so
So
it's
it's
Matteo
basically
Lab.
MatteoLab.org, yeah, .org. And then you will see there's a donate, should be really visible.
Okay, and I'll try to put it in the chat also when I'm not talking. Okay, we have another question about someone who has a lot of beautiful live oaks on their property. It's actually Jennifer, and it grows really fast, and so does bay laurel. And she's thinking it sounds like she needs to try to remove them, even though they resprout. She's saying she'll keep working at eliminating the bay laurel, but do you recommend anything for her oaks?
Oh, no, you know, I wouldn't be the person to talk to. I mean, I know some tricks about taking out bay laurels that we learned manually, but for the oaks, you know, I don't know. But certainly, the less competition you give them, the better. So, you know, they'll do better without the competition. For bay laurels, my suggestion is to pick the area. You know, because they sprout back. If you just thin them everywhere, you're gonna have a huge job ahead of you because they resprout and they'll be worse than they were in a couple of years.
Oh no.
But normally the best thing to do, think of your property as a patchwork of habitat, and then look at where you already have so much bay laurel and just decide that's going to be the bay laurel portion of the patchwork. And then find the areas where you have less bay laurels and more oaks and decide that that's going to be where you're going to help the oaks. So that's going to cut down your labor, your efforts dramatically. And you just have to let it go. You have to let the other patch go and let's take its course and put all of your energy where you have fewer bay laurels and more oaks. It's a suggestion that's been a winner over and over. And people come back to me and say, oh my God, we should have listened to you.
When you say put your energy, you mean you just keep going back and keep taking them out, keep
Right.
taking.
So you keep, and then also, I mean, you should talk to somebody who's an expert on this, but I can tell you that the time to, once you located the area where you want to do, and again, don't locate an area that's riparian or inside, you know, at the bottom of a draw, because those areas are really ideal for bay laurel. So we shouldn't really be removing them from those areas in the first place. So make sure that it's not one of those areas. Make sure that you don't have too much bay laurel because then it becomes impossible. Make sure that you have oaks as well growing. So once you pick your area, the best time for bay removal is the fall. If you are okay with applying herbicide on the stump of the bay laurel. So that is really, so do it don't do it in the spring because your effort will only give you 50% success. If you do it in the fall, your success rate goes up to 85%. But you have to cut and then you have to apply the herbicide on the stumps, sometimes the little stumps.
And then do you have a recommendation for a type of herbicide?
Yeah, the one that really works is Garlon.
Garland. Okay. Garland. Okay, I'll probably try to send that one out too. Okay. So let's see. A question came up. Once a tree is infected, how do you help it survive?
Yeah, that's a big question. So in our opinion, so when we did our 20 years ago, we did our first treatment studies because people really wanted to save their oaks. Before we had figured out that really the biggest thing you can do is remove bay laurels. That's the most important thing. So, but we developed this chemical treatment. One of the things that we learned was that the treatment was really effective only if the oak is treated preventatively. So if if you're treating the oak before it's infected. So ideally we tell people, hey, you know that your bay laurels are infected. This is the time to treat your oak. You know, it's perfect because you know you have infection. So it's timely, it's appropriate, it's legitimate, everything you want to call it. So that's the best time. However, when the tree is infected, bottom line, because I wanted to get a treatment approved in California, and it is, the data all of a sudden doesn't make a lot of sense because the treatment it's almost always effective. if you treat it preventively, but it becomes, random when the tree is already infected. And we think the reason for that is because the pathogen can move so quickly that by the time you notice it's infected, the infection is very widespread already. And so no matter what medicine you give it, it can't come back. And unfortunately, because we can't really tell visually, it's very difficult. So we don't have, I can't tell you, oh, if the tree looks like this, use it or not. What I have told people that if they have oaks that they absolutely love, and if they have oaks that they look at every day, so let's assume you have an oak in your backyard, and you look at it every day. And let's assume that all of a sudden you see the bleeding spot that's diagnostic of sudden oak death on the trunk. So you see this, sap coming out without any wounding and without any insects. So you see some gooey stuff, dark red or black, coming out of your intact bark. That is the first sign possible of Phytophthora morum. So that is a situation where I would say treat the tree, because yes, it's not preventative anymore, but you looked at this tree every day. So you caught the symptom as soon as humanly possible. So that would be the only, and still I don't guarantee it's going to help necessarily, but it's probably worth the effort. But if it's if you have a property and you never walk in your backyard, one day you walk after a year and you see it treated as a sap on its bark, and you treat it, I cannot recommend that, because you have no idea whether that, then you're just, and it's not what the treatment is for. It's really for preventive treatment or for early treatment.
I see. And can you remind us again, what's it called? What's the...
Well, so can't really tell you the names. There's several products now, but the compounds, it's called either phosphonic acid, phosphonic, for phosphoric acid, or phosphonate.
Okay.
And if you do a quick search, you do Phytophthora, phosphonic acid, you get a list of products.
Okay, thank you. A different topic, more related to your wider spread knowledge. The Mediterranean oak borer is affecting white oaks. Are you studying this as well?
Well, this was a failure of mine, because we started, we are the first people to get on, I don't study insects. Okay. So it makes sense that I'm not studying it, but I could have diagnosed a lot sooner, but unfortunately the entomologist at Berkeley was not very collaborative, and I didn't really know how to identify beetles. So I know it was a bark beetle, it was an ambrosia beetle, but I didn't know it was the French beetle. And so I decided to study the fungi they carry. And so to see whether the fungi had a role, and it doesn't seem like the fungi have a major role in killing the tree. So we think, but some role, yes, but not major. So really, it's the Mediterranean beetle that's doing it. Most of the killing directly. And again, I don't really focus on insects per se, so I'm not studying it.
Well, we appreciate everything you're doing with sudden oak death and all the other things. So that's
Yeah, I'm
great.
studying almost every disease in California, except for I don't study pests, because entomologists are better suited at doing that.
Okay, great to know. So how is the funding cut as of July 1, 2025 going to impact your research?
Major impact. So that's why I have to basically not shut down, but downscale significantly. Most importantly, when when your major federal funds get cut, the only way you can make ends meet is by firing the people that make the most money. So I was forced to fire my lab manager that had 40 years experience in the field, and my other lab manager had almost also 40 years experience, because the only way for the lab to survive was to cut those two people and just work with very qualified young people. So I'm very happy I have a couple of really qualified people, but that knowledge that these people had working with me for 40 years, one, and the other one 30 some, it's irreplaceable. And so as a result of that, a lot of the services we offer, like we used to do tree hazard assessments. We're not doing that because we don't have the capacity to do that. A lot of other things. And Yeah, so we're at the bare minimum. Even the soil blitzes have been scaled down to about 50% of the effort. So we don't ask people to collect less. We just process 50% of what they give us.
I see.
Yeah, with a method so that we kind of we make everybody happy. You know, we
Mhm.
don't say, because some people collect up to 20 samples in general. That's the average, 10 to 20, and they want to see where where the results of their trees. So if you collect it from 10, I would process only 5 of them and spread out in your collection so that they will cover the geographic area that you covered when you were doing the survey.
Okay, thank you. Thank you. We're really sorry to hear that and
And we may have to move a per pay where people may have to pay per sample.
Mhm.
The blitz. I'm trying to stay away as much as possible, but the large land ownerships, the ones that do hundreds of trees, they're already paying. They're paying half. Yeah, they're paying a smaller rate than what I would normally charge. So they have a benefit in participating in the blitz. And then all the private landowners, they don't pay by only process half. So in the future, there may be an option to participate and have all of your samples processed if you're, a landowner at a fairly small cost compared to what you would have to pay somewhere else.
Okay, I'm going to ask one more question and then I'll ask you if you have more time, but we're nearing 5:30. One of our hike leaders from around in the Bay Area is asking, is there more infection along hiking trails, and specifically are hikers a vector of the pathogen?
Yeah, so there was a published paper that found out that there was more infection on the trail. Actually, they were sampling the soil on the trail and then sampling the soil off the trail. And we never, I mean, we should have. So the problem with doing that is that the soil on the trail is much more mineral. There's less. You don't really have an old layer that's fully developed, and so Phytophthora doesn't have a lot of competitors on the trail. So the reason why we're finding more Phytophthora on the trails is not because people are moving it, but because as I said, this is not a good soil fungus. So this is a Phytophthora that has moved away from water and soil and now uses the air to spread. So it does really well on the leaves, but it's not a good competitor in the soil. So every time you have a trail, you have a somewhat better environment because the microbial community on that trail is simplified compared to the microbial community of untouched land that's next to it. So bottom line, we don't have any evidence in the Bay Area in general, but in California in general, of hikers really significantly moving the pathogen, 0. We do have evidence of, so it doesn't mean that it's not happening, but what we do find out is that the soil needs to be inoculated all the time. So the pathogen doesn't survive very long in the soil. And so that means that the soil, it's really not a very good medium for the pathogen, but it's not 0, so the soil can be infectious. We do have evidence that people moving infected plants, probably sticks for wildfires. I mean, sorry, for campfire wildfires. For
Mhm.
campfires have moved it. We have some evidence, a couple of instances where we have a sneaky suspicion because the first plants to be infected, they were near a campsite, actually near the individual spot, one spot in a campsite, had an infection right next to it where people were doing, would have done their fires and cooking and everything, and that's most likely where they put the wood down, mostly with foliage, So we do have some evidence, not very solid because, 2 cases, statistically, it's almost insignificant. And we have almost 0 cases of hikers moving the pathogen around.
So speaking of the hikers, how long does it survive on our hiking boots if we don't rinse
Yeah,
out?
well, it will survive. Again, it's a little complicated. So let's say that you go and you hike in the peninsula, right? Somewhere in one of the parks. And then and especially during I would say seasonality is key here. So if it's raining, if it's the rainy season, especially if it's in the spring, so if it's May and June, um, there's so much spores in the air that a lot of them end up in the soil. So the soil is really full, of spores, which makes transmission easier. It seems that by the time you've walked, especially if you walked, it's that pathogen, it becomes harder for whatever remains on the shoe for the pathogen to survive. And maybe we just don't notice it because the infection is everywhere in the Bay Area. So if it was a rainy season and you walked with boots in a peninsula and then you flew to New Zealand, then I
Mhm.
strongly recommend that you clean your boots.
Yeah.
Because again, it's not a plus minus. For sure, the soil can start infections. I just don't think we have seen it because the pathogen moves where people hike, the pathogen had already arrived and it already moves on its own. So it's not evident that people are moving the pathogen with the soil. But again, we tell people, well, you go away from the coast. And that's why looking at the sod map is important. Go to sodmap.org, download the map, look where the pathogen is. If you hike in an area with a lot of red trees, which means infected on the map, then really pay attention and clean. So I have a brush and I have some Lysol in my car, and
Mm.
even at the tails of the rainy season, I always clean my boots. If it's bone dry, maybe just brushing is enough, or sometimes there's nothing on the boots. And so it's really not necessary. So again, plus minus, yes, it's an issue, but in reality, epidemiologically speaking, we don't know. We have no evidence that it has happened. It may have happened. All the data we collected suggested that the pathogen had arrived on its own and through ornamental plants.
Oh, thank you. And I know everybody here wants to be responsible, so we appreciate the interest.
Yeah, I think the beautiful thing about Phytophthora ramorum, and maybe we can conclude with this answer, is some of you may be familiar with pitch canker of pines, another exotic disease that's causing a lot of pine mortality along the freeways. All the pines that you see dying is Monterey pine infected by pine pitch canker. And that pathogen, the spores are really microscopic, very, very tiny. You just don't see them. With the sudden death pathogen, the spores are really large. You can't really see them. You can almost see them with the naked eye. You can't really see them with the naked eye. But the spores need to be embedded in organic debris. So we found out that if you clean your shoes so you don't see anything, you don't have anything.
Oh, great.
Because the spore and the cocoon of leaf material or soil material is large enough to be visible. And so, bottom line, the most important thing, you don't have to worry so much about the chemicals. Really, what you have to worry about is, does it look clean? And make sure if you're like me, blind, make sure that you have your glasses on. And then, so that's really a huge benefit that we have, that we can be very conscientious and we can do a very good job without going crazy, just making sure that things look clean. Of course, if you have a car, it gets more complicated because you have the tires, but your shoes, maybe even the bike tires, tools, all of these things, if it looks clean, it's clean.
Okay. Well, hey, everybody, I really appreciate all the extra questions, and I'm sorry we didn't get to all of them, but I appreciate everybody's rapt attention to Matteo. And Jennifer, it looks like you're ready to say a few words.
Oh, no, I was just going to reiterate. I'm sorry we didn't get to all the questions. I think there was a little trouble this hour and a half with people getting their questions in the right place. And I promise the team will work on the glitches so that next time it's a little smoother.
Yeah, so I want to answer one question that's asked by Caroline Lacera. Did I understand correctly? You said Phytophthora doesn't spread through soil? No, most Phytophthora spread through soil. I'm talking about sudden death, okay? So let's be clear. I study a lot of soil-borne Phytophthora, and that's what they do. They spread through the soil. So when I say Phytophthora ramorum doesn't spread through the soil, it's the sudden death Phytophthora, it doesn't spread through the soil. I mean, it can spread through the soil, but we did a lot of epidemiological studies, and we don't get that evidence. And the soil needs to be reinoculated every year because the pathogen dies out during the dry season, and then it gets reinoculated in the winter by the pathogen that's growing on the leaves, which is very different from what happens with soil-borne Phytophthora. Sorry, that was a very good question. And it's very good for me to specify that because I don't want people to say, oh, Matteo said Phytopathology doesn't spread through the soil. We do. And water is another issue, water spread. And Phytopathology may spread through the water more likely than the soil. But then it's a hurdle for it to come out of the water. So you need a leaf that's right touching the water. So there's a whole process. And we think it may have happened a couple of times when we've had floods and then we had outbreaks show up after the flood. So movement through streams may be more likely, especially if you get a little, you get a flood, a local flood, that flooded area may actually start an outbreak.
Oh, right. That makes a lot of sense. Hey, just to be clear, I saw a couple, I wasn't sure, I tried to type in what you said about your donation lab, your lab. Can you verbally say that again and I'll try to type it in again? What it,
Well, if you go to matteog.org, there should be a donate button. And you just click on the button, you can donate the easiest way through a credit card. And then you're going to get a receipt that you can give your accountant or yourself, keep yourself to get a full tax deduction.
okay. Yeah, and Robin also put something in called the Matteo Garbelotto.
Yeah, that's it. That's
So the one I typed in didn't really work. So the one Robin put in did work, it looks like. So
I
the
think that's I
Garbelotto.
don't, yeah, I think that looks like the right number. So that's actually the precise link. But if you like images like I do, yeah, go to
Thank you, Robin. I really appreciate that. Thank you.
okay.
Okay, well, thank you, Matteo. Thank you so much for spending this time with us. It was just wonderful. We'll go ahead and get your PDF and we will send the link to the recording and the PDF out to everybody. We'll also post it on our website so that
Okay.
everybody can listen again and take action.
Okay.
And judging from the number of questions,
I know, sorry, it's
judging
not
from the number of questions as a parameter of the quality of the talk, this was great talk, very thought-provoking and informative. So I thank you too.
I appreciate it. So the website's actually matteolab.org, not Matteo G. It's matteolab.
Oh, okay. Even that one, for some reason, didn't work when I typed it in, but maybe I'm moving too fast. But you're saying the one that Robin provided is the right one. So everybody go to the one Robin provided.
But if you go to matteolab.org with the 2 T's, then it's very easy to navigate to the donate button.
Okay, let me try again.
It's on top. So if you do Matteo with 2 T's, lab.
Oh, I see. My computer didn't like it, but it did work.
Right. And then
I'll put in what
you can see donate. Donate is the second, on the front page, you can see donate. You click there and you'll get to you give now. So if you're using a phone, it's a little complicated because at some point you don't see anything. You got to click on the gift basket and that will move you forward to the next. On the web, you can actually click give it now and then you see everything. But on the mobile or handheld version, you actually have to click on the package. There's a little package in kind of master color.
And everybody, you can save the chat. You should be able to go up to the 3 dots in your window if you want to save all these links. You can go ahead and save the chat if you wish. If you want to go straight to the one Robin provided, or the two different ones Robin provided, either one, it sounds like will get you there.
So Matteo, I would like to thank you again for coming to talk to us. Everybody really appreciated hearing from you. And I'd also like to say that our next forum will be on July 21st, rather, the third Monday of July. We expect the speaker to be Bryant Baker talking about clear cutting.